Hello, my name is Juliana Restrepo Sibaja. I'm a third-semester med student in the University of Juliana, and today I'm going to present the effect of mother prenatal ethanol exposure on the differential expression of two histamine H3 receptor isoforms in different brain regions of adult rat offspring. So, first off, we have to talk about what H3 histamine receptors are. So, the histamine H3 receptor was initially described as a presynaptic autoreceptor controlling histamine release and synthesis in the brain. So it can have different effects on our bodies, like causing blood vessels to dilate, bronchoconstriction, blood clots, gastric acid secretion, adrenaline being released, increasing the permeability of the capillaries, frequent heartbeat, swelling, and inflammation. Yeah. So we have to have some concepts clear for the complete understanding of this study. So in the subject of prenatal ethanol exposure, we have fetal alcohol syndrome. Fetal alcohol syndrome are a series of conditions that are caused by in utero alcohol exposure. So it can cause a lot of phenotypical conditions that can be visualized by simply observing the subject. So, for example, microcephaly, small papillary fissures, smooth philtrum, thin upper lip, micrognathia, or minor ear abnormality. How can the alcohol ingested by the mother affect the baby to developing? So, alcohol crosses the placenta. This means that the developing fetus is exposed to the same amount of alcohol blood levels as their mother. This can directly affect the liver of the fetus developing, which is used for the growth and for red blood cells production. As a general objective, we have to evaluate the effect of mother prenatal ethanol exposure on the differential expression of two histamine H3 receptor isoforms in different brain regions of adult rat offspring. For this, we use different materials and methods, like the breeding of H3R knockout mice. Knockout mice are genetically engineered mice in which a single gene is manipulated. In this case, they carry an altered copy of the HRH3 gene with neomycin resistance, because neomycin is an antibiotic that can alter the blood alcohol levels or the liver failure due to these blood alcohol levels. RNA extraction and QRT-PCR from non-frontal cortex. In a PCR test, a specific genetic material sequence is amplified. In this case, we're working with RNA in a quantitative form. This is because we're testing the blood alcohol levels on the fetus through certain receptors. These receptors are obtained through proteins, and these proteins through RNA. Next, we have in situ or ISH hybridization studies. ISH studies make possible the detection of a specific nucleic acid within a sequence in a cell. So, the sequence is bound in a tissue section by complementary base pairing with a probe, which is a DNA dyed with fluorescence that is matched with their complementary base. In this investigation, the probes were designed to recognize RH3A and RH3C mRNA, which are the isoforms or RH3. Western blotting, which is a technique used to separate and identify a specific protein within a blood or tissue sample. In this study, we used it to detect protein concentrations in different areas of the mice, like P2 membranes of HEK cells or frontal cortex. Now for the results, we have figure 6 and figure 7. We have different variables, which are N for molecular weight, HEK for the cells that are being tested, and F for the isoforms of the HR3 receptors. Then we have a yellow band, which corresponds to the overlap of the recognized protein at 48 kilodaltons, and the flag tag being positive. In B and C, we have red bands, which correspond to the RH3B and RH3C isoforms correspondingly, and in D, a green band, which corresponds to the protein being recognized at around 48 kilodaltons. It was also recognized that there's a presence of the non-functional RH3D protein, but the molecular weight is too low, so it's not being represented here. The same happens at RH3F. And in E, it could be the same reason, because there's no bands showing. In figure 7, we have A and B. In A, we have a green band for the gen-script antibody and a red band for the reference protein beta-actin. Then we have M, which is the molecular weight market bands, and there's no 48 kilodaltons H3RA antibody sequence. As we can see here, there is no extra bands. In B, we have the comparison between frontal and cortical membranes from rat, wild type, and knockout mice. So there's no significant difference between the evidence of H3R protein, because as we can see, the Western blot is practically the same for both. Then we have figure 8 and figure 9. Figure 8, we have wild-type mice, wild-type rat, and KO mice. In wild-type mice and KO mice, we have a black square signaling 55 kilodalton band of a protein identified as beta subunit of ATPase. There's no MH3A protein detected as well. In figure 9, there's A, which is the chromatin-stained gel corresponding to the B Western blot, in which we can find the molecular weight marker lanes. Then we can find that 20, 30, and 50 are the amount of micrograms applied of P2 membrane correspondingly. So the yellow bands represent the H3RA antibody binding to RH3A protein in all three lanes. At various kilodaltons, protein is it's bigger. So at different weights, for example, 55 or 70, we can maybe infer that there are dimers or even trimers of this same protein. The white box in both panels is signaling RH3A protein without the limit of quantitative reliability. Okay, so for our discussion, we had different authors' perspectives on similar topics within the investigation. So Moritz said that there are cases where there are either none or even negative correlations between mRNA and protein expression data. Disagrees with the study, and Bereschin said that given the limitations of interpreting changes in mRNA expression relative to measures of H3R function, it was important to quantitate H3R isoform protein expression in brain regions where PAE altered H3R mRNA expression. Disagrees with the investigation as well. Finally, Shenton et al. And Marcus Gomez said that consistent with our observations of Rh3A mRNA overexpressing HEK293 cells, a number of studies have reported that H3Rs can dimerize with a variety of other neurotransmitter receptors. This finally agrees with the investigation as well. To conclude, we state that alcohol is a dangerous substance that can cause various disabilities in utero, especially neurologically wise. Even though there aren't protein expressions that can firmly support this statement, there are phenotypical expressions and general symptoms that may be related to alcohol exposure in utero life. PAE is in some aspects sex-related. For example, alcohol exposure affects different brain areas in female and male rats. This may vary because of their sex, their chromosomal composition, or other variables. Even if prenatal alcohol exposure, which is PAE, doesn't affect H3R expression directly, it affects different mechanisms that will end up modifying its function. So it directly doesn't modify its expression on certain brain areas, but the continuous alcohol exposure may affect other functions of the brain that can affect the baby's life overall.